Okay, hello everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Roger Tootel. Dr. Tootel received a PhD from the University of California at Berkeley in 1985. Following a postdoc fellowship with Dr. David Hubble at the Department of Neurobiology at Harvard Medical School, Dr. Tootel joined the faculty there, and during that early phase, he studied the neural organization of the visual cortex in animal models. In 1992, he moved across town to begin study and develop fMRI studies at the MGH and Harvard Medical School Martinos Center. And since 2015, he and his collaborators have been studying fMRI responses in small cortical columns, which operate by quite different principles compared to the much larger scale response in conventional fMRI. I would just like to remind the audience to please address any question you have using the Q&A box or raise your hand. Dr. Tootel, thank you so much for coming here today. The virtual stage is all yours.
Well, thank you very much. It's nice to be back at the center virtually. In studies of personal space, the quote that most people use as sort of a default is, always in the introduction of their papers, is say, personal space is the area that individuals maintain around themselves into which others cannot intrude without arousing discomfort. So in the initial experiments that I'll talk about, we're just going to intrude on subjects or virtual subjects and then measure discomfort. But then later, we're going to get into the physiology of personal space, about which almost nothing is known in humans or even in monkeys. So first, I'm going to show you just a clip of, so you get some idea of what. See, the things are involved. Let's see. Okay, so just look at the.
How about you, Jerry? I'm smart. Examine the upward plans. Maybe I'll try and catch up with you.
Okay, so that was just to give you a sense of, what personal space is and what happens when it's violated. Like you can sometimes find this out, see this in cocktail parties or elevators or stuff like that. It can be really striking.
She wants to say hi.
So here's some guy who just hooked up this is sort of a complicated way to regulate personal space. Most people just, move away, and that's what you can measure. But this guy has it down. So these are just general points. Most of the rest of the talk are slides and pictures. So, but the field of personal space research had a boon in the 1970s and 1980s, and then it sort of went away. And recently it's sort of coming back, but at no time did anybody really dig into what the brain mechanisms are. So anyway, people have speculated that personal space may be related to the fight or flight responses, which people studied years ago in predator and prey species. Now, roughly, personal space corresponds to that distance within arm's reach. And that sort of implies that, that it has something to do with defensive behavior or perhaps grabbing things that are attractive. But the evidence mostly just talks about defensive behavior. And I was attracted to it because you can quantify the inputs and the outputs, and then hopefully the intermediary cognitive mechanisms. And so we're going to focus, when we get to the physiology, we'll focus on the parietal cortex, where there also is coincidentally a threat sensitive circuit that's been mapped out pretty well in monkeys, but nobody knows anything about it in humans. So this shows that personal distance is typically measured using a stop distance procedure. And there's sort of two ways you can do this, but the measured personal space turns out to be the same. So you can have a subject, an experimenter, and the experimenter walks towards the subject until the subject says stop, hence the term stop distance procedure. And then you measure what the distance is. This is down here in centimeters. Or you can have the subject walk towards the experimenter and stop where they begin to feel discomfort. And again, these two measurements, no matter how you do it, come out pretty much the same. And at a group level, you know, like a lot of people come to the topic of personal space thinking of higher order factors like, you know, culture and the level of intimacy and gender and height and all kinds of other factors. There's clinical factors too. Autistic people and people with schizophrenia tend to have larger personal spaces. But here you can see the variation in personal space on average in all these different countries. And the US is, you know, in the middle somewhere, but it varies in different countries from like 80 centimeters to about 135 centimeters. So, and then as people get more used to the strangers then and become more intimate, then the values shift to the left. They become, the personal spaces become smaller. So, but despite this variability, within a given subject under equivalent conditions, personal space is highly reliable within a subject. So if you just use the stop distance procedure, you can find that, you know, subject 1, for instance, was relatively constant at 100 centimeters. And subject 6, for instance, was relatively constant at 40 centimeters. So there's wide variability in the sort of default personal space that people adopt, but it's reasonably stable over time. So this shows even more stability over a longer term. This is the baseline, it's the mean from curves like this. And you know, in the first session, it's about 70 centimeters. But then more than a year later, you can bring these subjects back and then measure them again, and it's almost the same, statistically indistinguishable. So that's kind of interesting. And it's because it's so stable, that makes it easy to measure, as you'll see in the subsequent studies. We used a single subject design because there's this variability here, but you don't have to worry about it sort of Heisenberg principle of just like fading away over time. You know, that would be hard to do an experiment on. So I've been talking about personal space as if it's like sort of a step function, you know, like there's near and then there's far, and then this is what we measure. And that was the original step function, but it soon became obvious, even intuitively, that if someone's really near you, you know, it's very uncomfortable. And the further they go, it gradually gets less uncomfortable. And then, you know, in what's called far distance, beyond the personal space boundary, then it's not uncomfortable at all. So different people have come up with different models about what that gradation looks like, but they're not known yet. And so here's one possible model. It's just a ramp. And then here's another ramp, you know, linear ramp. And then there's this one power function, just one possibility. And then one group claims that like there's 2 zones of uncomfort, you know, near and then far with the personal space boundary. So we went ahead and measured this, what's called permeability. And we found that it closely matches a power function. It's not the only possible function, but it closely matches. And so I'm going to talk to you about permeability, but I also want to talk about it in 2 contexts. One context is real subjects, like we've talked about so far. And the next context is in response to real, you know, human-like, but completely digital avatars in virtual reality environments. So you put these glasses on and, you know, you see some humanoid kind of person walking towards you or in the distance or something. And surprisingly, you get various forms of personal distance responses to that, even though you can clearly tell that, you know, there's a digital image. But because that's true, because you can get these personal space kind of responses, you can eliminate uncontrolled factors that are inevitable when using humans as stimuli. That is, you can do experiments that are absolutely exactly repeatable. You can extend the range of possible use of stimuli, you know, like missing or distorted body parts or flying stimuli or stuff like that, in a way that you certainly couldn't do with human. And I mean, every time I just discuss or propose disordered body parts, everybody in the lab kind of groans, but it's possible to do. And that way you could measure various different things. But in this talk, I'm going to concentrate on another advantage, which is that you can have avatars approaching you without worrying about COVID infection. So you can measure personal space despite the current pandemic. So this is how we measured both permeability and humans and avatars. So we just had subjects close their eyes, and then the experimenter would position themselves at different locations and then just stand there. Here you can see that the subject is at their own personal distance. They're 4 times as far away, and here they're within the personal space. And then we do the same thing with the avatars, and of both genders. We do it with women and men, and so that way we can measure personal space. So this shows the response to humans and avatars at different distances. So this is, you could think of this in centimeters, but these are all normalized to each person's personal space. So this is distance down here, normalized distance. And you can see that in the humans, which is the simplest to consider, the dotted red and black line indicate the data, and it forms a nice curve. And we tried to match it to an exponential or logarithmic or power law, and it fit the power law best. So, and the interesting thing is that the response to the avatars in terms of discomfort rating was almost the same as it was to human. That is, it started off steep. You know, people are always uncomfortable when when a human subject is near you, and then it fell off like a power function as the avatar and as the human went away. So one question these days is, what is the relationship between social distancing and personal space? So these personal space values are something like 60 to 100 centimeters, but the distance that we're asked to socially distance is 2 or 3 times that. You know, it's 6 feet in the US and 2 meters in Europe. And, you know, different countries have different distances, but it's always much bigger. So you're kind of stretching out. If you stand further away than you normally would, you're kind of stretching out your sense of what's appropriate to how important it is, how you want to stand. So this shows some of the data, and it's kind of cool. This is work mostly done by Sarah Zapetis and Daphne Holt. But it was cool. When the pandemic hit, I guess that was March of 2020 or something, for the first time, we were in the middle, or they were in the middle of measuring personal space without a mass or anything, because that was unknown at the time. So they'd measure personal space, and this is the mean they were getting at the time from, I don't know, 10, 15 subjects or something. And then after they were starting to socially distance by quite a long time, we found that the personal space size had increased. But one could say, well, you know, maybe they have some sort of generalized worry about an infection during the session that they're scanning. So then again, we did it in avatars. So this was the mean personal response to the avatars before the pandemic, and here it was after the pandemic. Of course, everybody that does this experiment is clearly aware that there's no danger of infection from the goggles. So that's kind of cool. Now, it will be interesting when the pandemic goes away, if it ever does go away, to see if these values go down or how quickly they go down. But it's interesting that, you know, that just social distancing in general has kind of affected what the norms of what we think of in terms of our personal space. So you mentioned that so far we've discussed the discomfort evoked by variations in distance away from a subject or an experimenter. And so we thought, well, if this is uncomfortable, maybe there's a sympathetic nervous system response that is, you know, that would show up in skin conductance measurements of the polymer skin conductance. So again, we measured the avatars at different distances from the subject, and humans, intruding humans at different distances from the subject. And otherwise, these conditions are measured exactly the same. And as you'd expect, as in the discomfort behavior, at far distances, shown in blue and green, both the avatars and the humans don't show much response, maybe a little. 100%, the white or the yellow, is again kind of near the border, it's the threshold of personal space. But then as you go nearer and even nearer in both the avatars and the humans, you get a higher response. And so again, this shows the function on the bottom left, shows the function in the humans and avatars. And again, it's like a power function, very similar to the discomfort ratings in the behavior. In fact, this shows both the discomfort ratings and the skin conductance ratings in the humans and the avatars. And you can see how closely they match each other, although you can argue that the y-axes are arbitrarily aligned, but still, it shows that the SCR may be reflecting discomfort in this case. Okay, so finally, now we're going to talk about the column scale imaging of distance or space encoding in human parietal cortex. Now we've chosen, well, and again, so here we're going to use functional MRI in the Martinos Center. We use the 7T and we use very small voxels. So we're trying to get at a size and a sensitivity where we can see columns if they exist, or at least high resolution activity. Because the columns, again, I don't know, it's hard to describe exactly, but it's like, normal fMRI is kind of like looking or trying to taste a stew or something. It has all these different tastes mixed together. But columns are sort of like, tasting the carrots and the potatoes, each of them individually. Like the columns, what you see with conventional MRI is just kind of everything all mixed together. But with the columns, you're seeing what are the computational units that the neurons themselves are, how are they grouped to function in ways that the general function just isn't known. So anyway, we measured responses to different virtual distances in the scanner using virtually and personal and visual stimuli, and I'll cover those later. So that gave us a baseline of the personal distance in each subject when sampled in real life and in the scanner. So there's two measures of their personal space. And our hypothesis was that in parietal cortex, which is just one step beyond visual cortex, we were hoping to find some reflection of personal space, this sort of power function, basically, in response to different stimuli. And we were hoping that that power function would have a personal space value that was similar in parietal cortex as it was in real life. I mean, in behavior. So now I've covered why parietal cortex. It's the next stage beyond visual cortex, and coincidentally, it's got a known threat-sensitive area in it. Like you can stimulate electrically certain parts of parietal cortex, and the monkey will act as if they're, fending off some invisible threat or something. It's kind of dramatic. And so I also talked about high resolution. So this shows our two measurements of personal space, both outside the scanner in the standard ways that I've talked about earlier. One person approaching another person and stopping when it becomes uncomfortable. And here we did the same thing in the scanner. Of course, in the scanner, we couldn't have a person approach, but we had faces approach. So presumably that's why there's more noise in the sampling, but you get the same means inside the scanner and without the scanner, if you equate for the size of the face in both conditions. So it was about 60 centimeters in both, and that's just their standard mean that we find all the time. Okay, so now, because we have the personal space in the subjects, fortunately they didn't differ, we're able to define the distance at which we're hoping to find a response in the columns that one might find in parietal cortex. So one way to sort of mimic the stop distance paradigm, and also just to, it's one way to evoke responses in things that might be tuned for approach or withdrawal, is to use a stimuli. It's called a looming stimuli. And this shows an example of that stimulus. Some of the time period would be like this. It's an approaching face, and its controls, we'd use faces like this. And the set of images in each condition are exactly the same. You know, I mean, ignoring the gender difference for the moment, which we equated for. But in one condition, in this approach condition, they're sort of intruding on your personal space, and you can anticipate that they're gonna, you know, you can anticipate a little bit that they're coming. So anyway, we found here and in many previous studies that this approach condition compared to the withdrawal condition, when both sets of images are exactly the same, except the timing of them differs, is reversed. You can see that in the folded brain, see, orange here is approach, orange and red, and blue, a little bit of blue, is the withdrawal. So in parietal cortex, which is here, there's a patch of stronger activity to the approaching phase. So to see it more clearly, we inflated the brain using the standard free surfer tools. And we were interested in the posterior part because that's the part that's nearest visual cortex. We figured if there is a transition between visual spatial encoding and this sort of personally distorted facial spatial encoding, it would show up in the inferior parietal cortex. And there's actually been a couple proposals to this extent in the literature. So anyway, the white is our region of interest. It's based on the sulcal gyral topography, but you know, it turned out that the activity, the bias for approaching faces was right in the middle of there. So this shows the same data in the flattened patch, where we cut off something like this, and then you can see again posterior bias. There's a posterior activity bias for the approaching faces. This arrow just points to a tiny little area that's interesting, but I don't have time to talk about. But all this is an approach bias. And the approach bias gets carried forward, but we're only looking at it here for various reasons. Okay, so this shows, no, I'm sorry. This is a group average of all the activity. So of course, you can't really see columns in a group average because they all mush together, they kind of average, and you get kind of a big patch. But if you look at the individual hemispheres, this is subject one, or one subject, left and right hemisphere. There's another subject, left and right hemisphere. The white is our region of interest based on the sulcal gyral anatomy. And you can see there's a lot of activity that's biased for the approach patches, or that's biased for the approach stimuli, and a little bit of activity in blue for the withdrawing stimuli. So you could argue maybe there's columns for both, and they, you know, sort of, maybe there's a push-pull mechanism or something. But anyway, at least we see distinct patches, and the patches, this shows a map of the patches in different layers. So this is our first test of whether the activity was radially aligned, like a column would be. So here you, anyway, just the evidence from the fMRI is if you sample the activity from a free surfer depth of near the surface, it looks like this. And then if you go down halfway through the gray matter, you see that the pattern looks very similar, you know, essentially the same. You know, this patch is this patch here is this patch, et cetera, et cetera. And it continues being similar all the way down to near the bottom. And then even when your center of the activity sample is, or where you're centering the map is half white matter, and then the rest gray matter. And that's why it's dimmer here. It's not as significant because half of it is, you know, partial volumed into the white matter in which there is no activity. But this strongly suggests doesn't prove, but it suggests that we may be looking at a radial organization in the bold patterns. So does this mean that there are approach columns and withdrawal columns? That's what we're going to test next. But just as background, so you can understand clearly, I mean, a lot of you will understand what a cortical column is, but if you don't understand it, the cortex is a sheet of gray matter. And here we've flattened it out using either computational methods, or in the past I used anatomical methods. And this shows the columns from a surface view of the flattened oculodermis column. So this is in primary visual cortex, way down at the bottom. And these are probably the most well-known columns in the research world. Anyway, so if you open the right eye and then close the left eye, you'll see columns in primary visual cortex. The gray is regions that were more activated by the open eye. The white is regions that are activated by the closed eye. And the whole activity was labeled with deoxyglucose, which is not a technique used very often, but it has high resolution. And you can see that the columns are very fine scale. Like this is a 2 millimeter scale bar. So this is partly why, you know, it's a challenge, but a worthwhile challenge using fMRI to try and see these columns. So from a view, this is a view, like I said, of the surface. This is a view taken perpendicular to that, showing, you know, top layers and the bottom layers. And you can see that the activity is radially elongated. So that's one of the defining properties of columns. So we're going to try and see these things and see them in both views. Okay, so this shows how we're trying to get from a surface view to the side view that I just showed you. Like this is surface view, this is a side view. So this shows kind of a zoom up of one of these activity maps where this patch responds best to, this is a surface view, this patch responds best to the approaching stimulus. These patches respond better to the withdrawing stimulus. And I've deliberately drawn a sampling line between them to see what they look like in 3D. So you can skip this as sort of a recalibration of the color scale. If you go from this to this, we're just turning it 90 degrees. So we're seeing it on the side here. You can see that this patch is radially elongated in cortex. That is, here's the cortical surface, here's the white matter down here. And you can see that the activity is kind of radially elongated. Again, here's this patch of an approach patch. And you can call it an approach column because it really extends radially. And the same thing for here. This is just an example from somewhere else in cortex, just showing the radial elongation, but I'm not showing you the rest of the slide. Okay, so what I showed you was just examples. You know, you could make up a story and show your best work or your best images and then claim that it's columns, but we wanted to do it in a more systematic way. So we analyzed the functional variation in humans that have looked at the approach versus withdrawal stimulus. And we looked at the activity in each vertex and we sampled them either radially, you know, from the cortical surface down to the bottom. And then we just added all the vertices together and see how well they correlate with each other, like from here to here or anywhere along here. And then we compared that to sampling in a cortical surface or parallel to that. So we sampled here, for instance, in the upper, and as a control, we sampled in the lower. And we're just looking at the amount of functional variation along this and along this. And it really supported the radial model, that is that these were columns. So in the following way, this shows the correlation between vertices along one of these samples. And in the sampling across columns, that is in a surface parallel view, you got a correlation between vertices of, you know, 0.3 to 0.4 or something like that. But if you're sampling radially within a column, a presumptive column, you know, from the surface to the bottom, the functional activity was much more similar to itself, to the other vertices in that column. And the correlation was 0.72. And those values matched the correlation differences in a previous study we tested in an area in V2 in monkey that is known to have real columns. So this was even stronger. So it looks like these patches are columns. So now one control experiment is, the first experiment we did as a control is that the approach and withdrawal columns move all the time. And so you could say, well, is it an artifact of the fact that the motion is either towards you or away from you? Whereas personal space, somebody can just stand there and it'll still produce discomfort or not, depending on the distance. So we were wondering whether people are responding to the distance variations, if you imagine these to be stationary heads, or whether it was the fact that they're moving towards you or moving away. So in panel B, what we did is we presented these different faces of different size and 3D cues. Like it's basically different virtual distances, but they were stationary when we presented them. They weren't moving. So this shows the response when you sample within the approach and withdrawal columns, shown here in cyan or red, and blue. It shows that outside the personal boundary, there wasn't much difference. They are all kind of flat. Here's 0 here. Whereas here's the personal boundary, and after that, the closer faces produce higher and higher responses in a nice monotonic curve. On the other side, the cortical regions that were responding to the withdrawal patches, it wasn't quite as clear cut, but remember, these are the most, the dominant kind of columns in parietal cortex. So that looks sort of like our power function in inverse, because this is inverse. Okay, now the second control experiment is a little more complicated. These faces, or all the face stimuli that we use, both the stationary and the moving, they all vary in 3 dimensions. That is binocular disparity cues. Like if you go to a 3D movie in real life, and what you're looking at is images where basically they vary the binocular disparity. That is, one image is the one you would normally get in real life to the left eye, and one image is the image you would get to the right eye. And the brain, magically puts this together. And if the disparity isn't too great, then you'll get the impression generated by the brain that this is 3-dimensional. So anyway, in visual cortex, further posterior, it's known that some cortical columns do respond preferentially to either near binocular disparities or far binocular disparities. Those that are activated by near distance and far distance. And so we wondered, is the personal space, are the personal space columns an artifact of stimulation with faces at different disparity? And you could argue a priori that this wouldn't work anyway, but we decided to do a test for that. So we tested 3D based near and far columns using disparity cues unrelated to faces. So this just shows how such experiments are done, and you have to have a leap of faith here. This patch of random dots is presented to the left eye. This patch of random dots is presented to the right eye. And perceptually, these appear, because some of the dots match and some don't match, it looks kind of like skyscrapers moving near or far to you. This is another control where they're matched up and they don't look like anything. So we're preferentially activating, using these non-face stimuli, we're preferentially activating the columns that we know to exist in visual cortex that will respond to near stimuli. And then in other parts, in paired parts of cortex, they would, different set of disparity columns would respond to far stimuli. And so this shows the previous example I showed of the approach versus withdrawal columns. And in the same subjects, that is subject 1, left and right hemisphere, subject 2, left and right hemisphere, you can see that we got, both of them produced orange and blue patches. In this case, they were approaching versus withdrawing. And in this case, this disparity stimulus also produced near and far patches, orange and blue, but in this case, they were near and far in the disparity. And so you could look at this and think, well, maybe they're the same, maybe they're not, who knows? Does this patch match this location? And, anyway, you can't tell by just looking. So, and that's our hypothesis, is that these columns are different from these. So they'll be in a different cortical location. So here, there's 4 different possible columns. There's the near disparity and the far disparity. And those are coded by yellow and green here. And then we overlaid maps of the approach versus withdrawal columns and the withdrawal versus approach columns. So there's 4 possible columns here. And if there's any overlap at all in them, we coded that black. And you can see that the black regions, there's not that many of them. There, it looked to us as if they're interdigitated, all these different kind of columns, which would be great, because it would mean that they're not the same thing, which means that it's not an artifact. So here we quantified it more by sampling the vertices, the activity in the vertices all around each of our regions of interest. And if you just shuffle and mix up all the activity, the biases, you get this, these black curves. But if you just sample it when it's aligned as it is in cortex, you get a systematic non-overlap. There's less vertices that overlap, shown here in the magenta here. So that showed pretty clearly that, anyway, this is our conclusion from it, that in cortex, you have 2 different kinds of distance encoding columns. One is personal near and personal far. That's, we diagram that here. And the other is disparity-based near and far. And so we think, and this model arose because of the systematic non-overlap of the different kind of columns. And so we think maybe they talk to each other. One is, it's as if the disparity information is coming straight from visual cortex. That's kind of linear, it's kind of straightforward. And then the personal space information is more, you can model it more closely by a power function or some fall off like that, which in the, at the personal space. The disparity thing, the disparity property doesn't even show up as a zero crossing, or it's just linear. There's no power function distortion to it. So this is the people who helped, and there's a lot of other people in Daphne's lab who also helped. These were the pictures I had. This is Daphne Holt, she runs the lab. This is Mona, whose actual name is Zara, I think. Shaheen Nasser, Aktash Abadi, and Sarah Zapetis. And Doug Grieve also gave invaluable help. John Polmini helped us get going. And Dylan Hughes also helped a lot. And also NIH helped a lot. Thank you very much.
Thank you very much, Dr. Tootel, for this great talk. And we now move to the Q&A phase. I'll just remind the audience to please address your question either using the Q&A, the chat, or just raise your virtual hand. And I think we have the first questions from Iris Chen. Iris, we're going to try and allow you to be panelist in a second. Then if you could unmute. And while this is being performed, I'll enjoy the benefit of asking a quick question. Did you, Dr. Tootel, see any differences between, I don't know, gender differences, a male being approached by a woman or a man, or vice versa? Or did you see any differences when different heights of avatars were used? Or did you look into that?
A lot of, most of the experiments in the 70s and 80s were of that type. We didn't look at it. We equated the genders, and we, either controlled for or didn't have differences in arm length and stuff like that. So we were sort of equating those lower level variables and chasing, the other questions.
Thank you. And Iris, can you please unmute?
Yeah, I'm here. Can you hear me?
Yes.
Yes, I can hear you.
Yeah. Oh, hi, Roger. I
Hi,
think this
hi.
is very interesting. So for the withdrawing and approaching study, so I suppose the parity study is using the non-phase approach already. But if not for the parity study, I just wondering that how big the phase occupy the whole space.
How big is the phase? Is that?
Right, and if you replace it just the approaching and withdraw with any object,
Yeah.
are you going to see the
there's
similar
two questions.
results?
Yeah. Okay. So the withdrawal and approach experiment was done in a conventional scanner in 2014. Holt et al. And so that shows you the group averages there. And they did test the effect of approaching cars or an approaching ball that had no context. And basically the faces produce much more activity. So that was comforting to us. Insofar as we were using electronic faces. You also asked about the field of view, perhaps, of the visual stimuli, if I understood correctly. And it was about 20 degrees, so it's semi-realistic, like the bigger your field of view, the more it's like real life. That's the principle, one of the principles in when you're using the goggles. We couldn't use the VR goggles in the scanner for technical reasons.
Thank you.
Thank you.
Great. Next question from Bruce. You can please unmute.
Hey, Roger, fantastic talk. As always, super interesting. Two questions, if we have time. One is the kind of it was interesting for me to see the laminar, if you will, analysis as a way to document column behavior, showing that in fact you would see something similar as a function of depth. That was the higher correlation that you saw, and that makes sense. And certainly consistent with the what say the FDG, the 2DG data showed. But of course, people are very interested these days in trying to differentiate behavior across different layers of the cortex. And in some settings, of course, there's reason to believe that there are laminar differences between input and output layers, et cetera. So where do you see kind of this use of similarity across the layers as kind of the relevant metric as you've used here? And does that tell us anything about our ability to be able to glean inference about layer-specific processing using RMR techniques?
Yeah, no, that's an excellent question. It's kind of, I mean, the difference between layers is often related to, there's not that much functional difference. Well, if there, I'm getting mixed up. The laminar difference is often related to the inputs and the outputs. That is, the idea is that you have one functional parameter which goes up and down the cortex radially. It's re-represented, like the ocular dominance columns. It's driven by the left eye on the bottom, driven by the right eye on the top, and but different layers tap out to other cortical areas. So you can infer the cortical connections from differences in the laminar function, but we've been focusing on the columns because that's where you'd expect the function to be the same up and down, but varying in the map. So if you tried to look at function in layers, I think Shaheen's chased this more than I have, but it's tough. We haven't been able to see any real laminar difference, even where that there's a laminar difference based on the monkey experiments.
It's interesting because to some extent your those correlations and the greater correlations in settings where you expect it to be columns does give you a sense that there is laminar specificity, not laminar, or at least depth specificity in the fMRI signals, or you would not expect to see a difference, right? If we really weren't sensitive to some degree layer by layer, then we wouldn't expect column structures to show a greater correlation than non-column structures. The fact that there is such a higher correlation in some ways is a hint that there really is some degree of depth specificity to the functional response. If it was just random, then it wouldn't look any different when you were in column structures versus other activated but non-column structures.
Yeah, no, exactly. But it's kind of important. Well, the key, I think, is that there's no conflict in having a functional variation between layers and a functional variation radially. There's no real conflict because they can be different functions, and they can be like you could have right eye dominance through an entire column, but what is done with the right and left eye information, when it's sent to the next area is different, and that may be reflected in a given layer. So I'm not trying to waffle here, but they're not logically conflicting with each other.
Yeah, and that's I think the important distinction, but it's not like it's either one or the other.
Right,
and when I look at your data, like I say, I'm encouraged because at least in those structures that you know to be columns and suggest might be columns, you're seeing behavior that looks different than other areas that aren't necessarily organized that way. And sure enough, as a function of depth, there's a high degree of correlation that that's not a universal across all functional activation. It's something specific in those structures where we expect there should be a column level organization. And we may not have resolved the issue of our ability to resolve input and output layers, et cetera, lamina, but at least it suggests it's not silly to be pursuing those questions based on the data that you do have.
Right. Yeah.
Just another quick question. I'm sorry. I'm sure there are others that want to jump in, but kind of you're thinking more broadly. So, I mean, you've moved out from occipital cortex, not a parietal cortex, into areas that are clearly kind of higher order processes like threat assessment. It's not just a primary visual stimulus and response. You've made that differentiation. Any speculation about column level organization throughout the cortex? I mean, if you start doing similar kinds of paradigms, in frontal cortex or, other areas, I mean, do you expect temporal areas, do you expect this to be a universal pattern, or is it completely unknown? What do we know? And like, what would be our next target of opportunity?
Yeah, no, I think, again, a very excellent question. In fact, essentially all demonstrations of columns prior to this, with minor, qualifications, have been in visual cortex. And so I think some people think, well, it's just a visual cortical thing, but you can imagine how useful this would be, given that cortex is kind of a two-dimensional or three-dimensional structure. If you could, to the extent you can nail these columns, because the functional specificity of the columns, all columns found, is much greater than kind of the whole ensemble of everything activating. But I think it could also be argued that there are columns in frontal cortex and places like that, but that people didn't use the right stimuli to elicit them yet. Because like who knows what those regions do? But if you could find columns, like just by chance, half the columns that are out there have been found by chance. Even the orientation columns in V1, Hewell and Weasel were just messing around, and the edge of their slide activated a cell that where if they turned the slide, the edge was different, and then they found orientation columns. I mean, first they found orientation selectivity, then they found it in the columns. But in the columns, these functional similarities, you can see them with fMRI, but we can't see single unit activity. But when a group of single units is grouped based on one function, you can see it with fMRI. And then, if you could do all possible stimuli in the world, you might, or all possible mental states or something, you could probably find the columns that way. But I think we're going to have to be a little more focused than that.
All right, that's great. Thanks. I really enjoyed the talk and super provocative.
Great. So I think at this point we can thank you once more, Dr. Tootel, for this great talk. And thank you everyone for joining. See you next week.